Enzymes is another class of membrane proteins that we can have. Of course, we know the definition of an enzyme. They act as a biological catalyst by reducing activation energy. The induced fit model is today the sort of most widely accepted model for how enzymes work. They don't have a fixed shape that is exactly complementary to the substrate. Rather, it's the proximity of the substrate to the enzyme that results in a slight change to the shape of the active site to make it complementary. The use of the word complementary is always usually underlined in mark schemes when you're describing any kind of enzyme questions. This allows the active site to mold itself to the substrate, and it forms a stable enzyme substrate complex. How does enzyme inhibition work? We've got two main classes, two types of inhibitors. One is a competitive inhibitor, or a non-competitive inhibitor. And they work in two distinct ways. And the naming here is quite telling of what they do and how they work. Competitive inhibitors bind to the actual active site. So what does that mean? It means that they will have a similar shape often to the substrate, right? Because otherwise, how could they bind to the active site if they don't have a at least partly complementary shape to the active site? So they physically block, they physically compete with the substrate for the active site. And so that makes a difference to whether they are affected or not by substrate concentration, because we can imagine that if we massively increase the concentration of the substrate, it will outcompete the competitive inhibitor for the binding. If there are more substrate molecules around, there's more chances for the substrate to collide with the enzyme and create a enzyme substrate complex than the competitive inhibitor, which is in smaller amounts. Non-competitive inhibitors, on the other hand, do not bind to the active site. They bind to an entirely different site of the protein, and they cause what we call an allosteric change. So by binding, as you can see here in this diagram, they will indirectly change the shape of the active site such that it no longer is complementary to the substrate. And so they will be unaffected by the substrate concentration. You can add all the substrate that you want. If the active site doesn't have the correct shape to bind the substrate, you've got no reaction. Let's see how that difference looks like. So these are quite sort of common graphs that you might be asked to interpret. So we've got our rate of our reaction on the y-axis and substrate concentration. This means that basically scientists will perform the experiment at a given substrate concentration, measure the rate. They'll then perform the experiment again at a higher substrate concentration and then measure the rate. And this is how these curves are constructed. And we'll often be asked to basically identify the shapes of these curves and deduce whether they come from a competitive inhibitor, an uninhibited reaction, which is the purple line here. So this is the uninhibited normal, I guess you can say, uninhibited reaction. And as I expect, my uninhibited reaction, as I increase the substrate concentration, so will the rate of the reaction increase until it reaches a plateau. What does the reaching of plateau mean? Something else is now the limiting factor, the amount of enzyme. So all of the enzyme active sites have been occupied here. So adding more substrate will have no effect. Just bear that in mind, because sometimes you might be asked to sort of explain the shape of this curve a little bit further in longer questions. In the green color here, we have our competitive inhibitor. And why do we have that shape curve? Because the Adding more substrate will increase the rate of the reaction. So the reaction can eventually reach the same rate as the uninhibited reaction. So solutions containing the competitive inhibitors are able to reach the uninhibited maximum rate of a reaction with a high enough substrate concentration. I'd encourage you to use that phrasing if you want in your answers, meaning that we can basically effectively drown out the effect of the inhibitor by adding enough substrate. However, if we are using a non-competitive inhibitor, like in this curve, then the reaction will not reach the uninhibited maximum rate of reaction because the active sites are themselves not able to bind the substrate properly. So adding more substrate is not going to help the maximum rate of the reaction. Let's see an example question. It's a relatively short one. Sometimes they can be a bit longer. Explain. So, of course, my command word here is explain. Sorry. Okay. Explain why non-competitive inhibitors are unaffected by substrate concentration. By context here is that substrate concentration and non-competitive inhibition, the relationship. My command is to give reasons, to explain, and my direction is to only focus on non-competitive inhibition. I don't need to explain anything about competitive inhibitors here. Non-competitive inhibitors bind to allosteric sites. Sorry, let me just. What's happening with the pen, so just give me a sec. Non-competitive inhibitors will bind to allosteric sites on the enzyme. That is one mark, just stating where they bind. So they don't need to compete with the substrate for the active site. That is my second mark here.